DNA extraction basically consists of four parts: DNA extraction, DNA binding, DNA washing, and DNA elution. First, for DNA extraction, 1 microliter of overnight culture was spun down to form cell pellets at 14,000 G for 1 minute, which were resuspended with 180 microliter of lysozyme digestion buffer containing lysozymes. The tube was then briefly vertexed before adding 20 microliter of lysostefin, 1 mg per milliliter, which was kept at -20°C in a fridge. The mixture was incubated for 3 minutes at 37°C. After that, 20 microliters of proteinase K and 200 microliters of genomic lysis/binding buffer were added before the tube was briefly vertexed and incubated at 55°C for 30 minutes. After that, 200 microliter of 96% ethanol was added, and the tube was vertexed for 5 seconds to obtain a homogeneous solution. Next, for the binding of the DNA, 640 microliter of the mixture obtained, or the lysate, was transferred to a spin column and centrifuged at 10,000 G for 1 minute at room temperature, 25°C. The collection tube with the liquid was then discarded, and the spin column with the matrix containing the DNA was transferred to a new collection tube. Following this was the washing of the DNA. 500 microliter of genomic wash buffer 1 was added to the spin column and centrifuged at 10,000 G for 1 minute at room temperature. The spin column was transferred to a new collection tube, and 500 microliters of the genomic wash buffer 2 was added before being centrifuged at 14,000 G for 3 minutes at 25°C, after which the collection tube was discarded. Lastly, for the DNA elution, the spin column was transferred to a sterile 1.5 ml microcentrifuge tube, and 50 microliters of sterile DH2O was added. The column was then incubated at room temperature for 1 minute by holding it near a Bunsen burner, and then centrifuged at 14,000 G for 1 minute at room temperature. The eluted 50 microliter of DNA was repipetted back to the spin column to recover more DNA, and was again centrifuged at 14,000 G for 1.5 minutes at room temperature. The DNA was then stored at -4°C in a freezer. All the procedures described above were done near the flame, as the pipette tips had to be sterile. After the extraction of DNA, the polymerase chain reaction (PCR) is conducted to amplify and obtain enough 16S segments whose two ends of the sequences are known. These are the conditions and sequences of the primers used for the PCR assay. After the PCR assay, gel electrophoresis was conducted to find out if the extraction was done correctly. 5 microliters of the DNA ladder and DNA of the bacterial samples mixed with loading dye were then pipetted into the walls of the gel. Once it was confirmed that the extraction done was correct, we purified the gels and sent it out for sequencing. The sequences of the bacteria were then blasted to compare the nucleotide sequences of known bacterial strains, and from there, bacterial strains were identified based on the statistical significance of matches.